Good afternoon, everybody. Thank you for joining this translational research seminar series hosted by the BSGCT in collaboration with LifeArc and the Cell and Gene Therapy Catapult. So firstly, some housekeeping information. You can see on the right of your screen here that to ask questions, you will need to sign into YouTube. You'll need to join the BSGCT channel, and then you can type your questions in the live chat window. Please include your name and affiliation with your question, and those questions will come through to me as the chair, and I will ask those questions to our speakers at the end of the session. So today we're on session 2 of 4. So last week you heard about the development pathway for gene and cell therapies from LifeArc, and this week we will be looking at the regulatory considerations. So this week I am welcoming Dr. Keith McLuckie from Catapult, who is head of non-clinical and part of their access strategy team. I also welcome Dr. Nick Darville, who is academic engagement officer at the Cell and Gene Therapy Catapult, who covers the UK South. So thank you very much for joining me. If you would like to share your screen and turn your camera on, I will officially hand over. So thank you.
Thanks very much, Carly, and thank you for joining us today. So my name is Nick Darville, as Carly mentioned. I'm going to start by giving a short introduction to the Cell and Gene Therapy Catapult, and then I'll pass over to my colleague Keith, who will speak for the rest of the presentation to give an overview of some of the development and regulatory considerations for advanced therapies. So do you want to move on to the next slide? I'm starting with this slide just to highlight the sort of growing strength of the UK advanced therapy sector. If you go to the next slide. The Cell and Gene Therapy Catapult was set up over a decade ago to support this advanced therapy sector and in the UK and also now further afield. We're an independent innovation and technology organisation committed to the advancement of cell and gene therapy. And really our overall vision is a thriving industry delivering life-changing advanced therapies. We work with academia, industry, healthcare providers through collaboration to develop new technology and address challenges for the sector. The Cell and Gene Therapy Catapult is comprised of teams of experts numbering more than 400 people in total. We've got unique capabilities and assets. Move to the next slide, please. So here you can see that the Catapult works with a range of stakeholders across the advanced therapy sector, stretching from later research and development and preclinical development all the way through to clinical adoption for advanced therapies. We can work with university or hospital-based advanced therapy research to support development of that research into translation and potentially initiation of a clinical trial, and we often do this via grant funding. We also engage heavily with regulators and maintain strong relationships with leading regulatory bodies or agencies. We've helped shape the regulatory landscape for advanced therapies, and later in this talk we'll be touching on the regulatory aspects. Next slide, please. So this is just a snapshot of some of our recent collaborations and partners, and gives an example of sort of the breadth of where we're working across the Catapult. Next slide, please. So on the left here, you can see our objectives. These are sort of overall objectives for the whole organisation, designed to overcome what we consider as key roadblocks for the advanced therapy sector. Then on the right is an overview of our capability, expertise, and areas of focus, and how that links to supporting key sector needs. I'm just going to quickly highlight a few of these capabilities that may be more relevant to academic advanced therapy research. Next slide, please. So first, we have our technology and process innovation groups and laboratories. These are internal laboratory groups. Through these teams, we can collaborate with partners to solve problems in process and analytical development for cell or gene therapies. And that can include aspects like scalability, cost of goods GMP preparedness or good manufacturer preparedness for manufacture, and robust cell or gene therapy manufacture and product characterization. Our internal labs have state-of-the-art equipment to enable that process and analytical development. And the teams have got a really varied skill set, including bioprocessing engineers, cell biologists, analytical experts, immunologists, and data scientists, just to name a few. We've built platform capabilities through these lab teams to help solve problems in these areas. An example will include a scalable end-to-end suspension-based AAV manufacture platform and a scalable iPSC manufacture process that's evolving to look at controlled, scalable, directed differentiation. Next slide, please. We also have non-clinical regulatory and health economics expert teams and the bespoke methodology that's been developed by these teams. So this type of expertise can help an advanced therapy developer with the route to the clinic, helping them to address challenges in preclinical development and translation, safety considerations, regulatory requirements, assessing commercial viability and reimbursement through health economics tools. Individuals in these teams have prior experience gained via previous roles in industry, regulatory authorities, and working within health technology assessment bodies. And the support provided by these teams is designed to be really integrated and holistic to help an advanced therapy developer plan a roadmap for development from a preclinical stage into and through the clinic, and then help that therapy developer meet the different steps and requirements needed to progress through the roadmap. So addressing those safety considerations, thinking about regulatory requirements, preparing for a clinical trial application, potentially helping to inform understanding around reimbursement and a clinical development plan. Next slide, please. So the final area I'm going to talk about is overall we aim to help de-risk a cell or gene therapy approach. We aim to make that approach more attractive to follow on funding or securing investment. We have a dedicated team supporting spin-out formation or commercialization if this is the right approach for a given therapeutic. So I'm now going to pass on to my colleague Keith, who will provide an overview of the development and regulatory considerations for advanced therapies. And thank you for listening to this introduction to the cell and gene therapy catapult.
Okay, thank you, Nick. Thank you very much. So now over to my stuff. Brilliant. This all may be a fairly daunting prospect to all of you. You've got your new R&D candidate, and you really need to think what you need to do to go through the translational process to get to the clinic. So the trick is really to plan your strategy, use the tools that can be provided in the industry, and above all, follow the science. So if we go through this slide setting the scene, ATMPs offer significant potential to address unmet medical needs. Enabling patient access to these innovative therapies in a safe but timely manner is critical to establishing their potential efficacy and value. Navigating the complex regulatory environment to obtain the required approvals to facilitate patient treatment can be challenging. So some aspects are similar to what you may or may not know from a small molecule biologics point of view, such as a monoclonal antibody, but realistically, there's a greater general complexity for anything in the ATMP market, and that essentially comes through in the work phases that you need to do. So depending on the product type, sponsors will need multiple approvals. So you have your clinical trial authorization, your CTA, and that is really what you're aiming for. You're moving your R&D phases all the way through to this, which gives you the go-ahead to put your therapy into the clinical phase. So that's first in human clinical trials. You may need to think about the ethics. For instance, if you've got some sort of embryonic stem cell, there's the ethics of using those sort of cells. If you've got some sort of cadaveric tissue-based product, there's ethical requirements for that. Genetically modified organism, so there's essentially paperwork that you need to go through from this point of view. So that's all about AAVs or any other gene therapy, you are manipulating genes, and that has to be essentially categorized. So there's tissues and cells. So there's the Human Tissue Act. So there's various different restrictions of what you can and cannot do. So, obviously from that point of view, there's approvals. And then finally, there's your local NHS R&D approvals. So these all come together, and these are the cogs that, click you essentially towards your CTA. So this schematic here is really, everything that you potentially need to worry about. So maybe take a picture. So as you can see on the top segment, there's 3 different workflows or work streams. So the lilac is really about the clinical phases. The teal is really about the safety or the non-clinical, and then the blue at the bottom is really about the CMC. So these work streams can run in parallel, and they all work together and they integrate as I will show you later on. And in the bottom section under the regulatory roadmap, these are essentially the pivot points and various different applications or documentation that you have to work on to dip into these work phases. So if we start at the left in ATMP classification, so you have your product, you think, well, what is this? How do I transfer this through to the clinical phases? So the first thing you really want to think about is ATMP classification. So this can be done with the MHRA or EMA, and really that tells you that it is an ATMP, and therefore you follow the ATMP guidelines. So is it going to be a cell therapy or gene therapy, or potentially a combined therapy, which is either a cell or gene therapy along with a medical device? And if there's a medical device involved, then you have to follow ISO guidelines as well. So that makes it a little bit more complicated. So realistically, the difference would be, have you got some sort of vaccine or some such blood product? And if that's the case, you may be better to follow those guidelines. So you've got your ATMP classified. You then think about, well, what do I really want in the clinic? So you may think about looking at a target product profile. So this is, or TPP. So this is a document that really you fill this in with a view to what your strategy is going to be, across the board to get you to the clinical phases. And so that document can really help you if you get involved early on. That is a document that really grows with your product through the stages. You then move into your clinical development plan and work with surgeons and experts to work out what you want to do in the clinical phase. And ultimately, you will have your clinical protocol. So if you come down a grade, it's the non-clinical development plan. So this is really proving that your product, whatever it is safe. So it's realistically going to be safe to go into patients. So it may come through in vitro, in vivo testing, and ultimately this is the documentation which goes into an investigational brochure or an IB, and that documents and justifies that your product will be safe to go into people. And then finally, the CMC or the manufacturing space. This is the final blue work stream. So a QTTP in this space is the quality target product profile. So that's really a document that really details everything to do with CMC or the quality. So manufacturing, CMC, anything to do with the production. You work on that, and then your CMC development plan, and ultimately you will then use these documents to fill in your IMPD, which is the Investigational Medicinal Product Dossier. And these 3 documents, the clinical protocol, the IB and the IMPD, help you justify your CTA or your clinical trial administration. So as I said before, in the lower part of this diagram under the regulatory roadmap, so these are the different points that you may need to consider. So obviously the CTA, that is the goal you're looking for, and the ethics that come with that. You may need a GMO approval. If it's a rare disease, you may need some sort of orphan drug designation. And realistically, if that's the case, we would say go for that, because that will help you down your cascade of work to get to the clinic. If you want to give your therapy to children in the clinic, you then need to work on a pediatric investigation plan, or PIP. Along the way, you may want to talk to the FDA or MHRA or EMA or whichever regulatory agency you need via a scientific advice meeting. So that is something that you really need to put into the calendar and work towards, because at the moment they are quite hard to get hold of because of timings. Then there's things, development support programs such as ILAP that you may or may not have heard of. So this is the innovative licensing access pathway. So this is really an accelerated path where you have various different tools. You can get hold of advice a lot easier than going separately yourselves. So you can talk to the MHRA, you can talk to NICE, you can talk to the NHS, and you can start with non-clinical data. And this really can potentially accelerate you to the clinic. And once you're in the clinic, it can accelerate onwards. And then there's things like the SME status and the legal entity requirements that you really have to think about. So working towards the first in human trial requires an integrated multifunctional approach. There will be elements of CMC, non-clinical and clinical development plans that feed into this strategy. Data generated during development essentially feeds into the 3 core regulatory documents required for CTA approval. So that is your IMPD, your IB, and your clinical protocol. And this data is also used to support the raft of regulatory processes that you must comply with, and development tools that you can utilize. So this is a fairly difficult to comprehend sort of workflow of what we're ultimately talking about to get to the clinic, but I'll try and break it down into some easier segments. So the first thing to think about is your documentation. So as you can imagine, there are lots of documents that you have to read or fill in to get you through to the clinical phases. So let's talk about risk assessments. So things as, risk assessment for your starting material, donor IDM testing, there's various different regional guidelines. Things such as adventitious agents, there's a European pharmacopoeial method based on that, the same for raw material risk assessments. And then there's things like excipients. You need a risk assessment to really work through those. And then there's also impurities. So all of these are really covered by ICHQ guidelines. So Q is for quality, and quality realistically means CMC, GMP, or anything to do with your manufacturing. There is then also ICHS guidelines, and ICHS guidelines are really to do with your safety. So the non-clinical phases, the testing, the efficacy, and everything coming forward to your GLP pivotal study that justifies that you can put that into animals. ICHM guidelines are all about what you need to get into the clinical phases. So Q, S, and M are the guidelines you need. And then there's also EMA and FDA documents that are really specific to ATMPs that really help you work out what you need to do. Obviously, you can potentially come to organizations such as Catapult, and we can help you through these different decision points and these documentations. But essentially, they are available to yourselves should you need them. So, technology selection. So, what is your manufacturing scale needs? You will start off with your lab grade R&D product, so that's small scale. Ultimately, to get to the patients you need in the clinic, you need to bulk that up. So you need to go to a medium and then a large scale, and it needs to be appropriately clinically GMP compliant. So this GMP compatibility, I think GMP is quite a scary word when you're in an academic lab, as is GLP or GCP. But this is really about sensibly making sure that everything is safe to move forward. So you don't necessarily need to be worried, you just need to think about it. So there will be a stage in your production where you have to be GMP compliant, but right at the start, anything to do with efficacy or proof of concept, don't worry yet. And regulatory compliance, there comes a point later down, essentially halfway to the clinical phase, where that becomes important. And I'll go through that in a minute. So your clinical use of your product. So you need to think, essentially, you will know what your product is, you will know the indication. So you need to make sure that the way you deliver that product helps with that indication. So if it's something to do with Parkinson's disease or Huntington's, it may well go into the brain. If it's something to do with, liver cancer or pancreatic cancer, or CAR-T, for instance, if that's possible, then it may be a direct injection into a tumor. So you really have to think about what your indication and the route of administration for that is. So in which case, there's implications of infusion, injection volumes, what cell concentration you will need depending on the target. And when you're talking about a gene therapy, obviously there's issues for that as well. And then from a point of view of handling, you need to make sure that there's the appropriate handling at your clinical site, and also when you do the testing in the preclinical phases, that your handling and your administration are similar to what you want to do in the clinic. So data packages to support CTA CMC requirements. So generally speaking, this is all quite dry, the CMC requirements, but ultimately PCQA is potential critical quality attributes. So these are things such as host cell protein, adventitious agents, and if you're talking about cell therapy, it might be non-differentiated cells. Impurity characterization, where required, you need to document that. So in the case of a cell therapy, it may well be non-differentiated cells again. Processes, so you need an aseptic process validation before you put this into patients, and largely before you put it into animals as well. And because of that, you will need batch data from a clinically representative process. And this is something that, as if you go through the cascade in a sensible, diligent manner, this will become easier to fathom essentially as you get towards that stage. So from an analytical point of view, you will need CMC type techniques to characterize your product throughout the processes of manufacturing, and to quantify and qualify and characterize at the end of production. So to begin with, these don't need to be qualified. As you go further down the cascade, they need to be validated. But either way, you need to make sure that your methodologies are verified throughout the procedures. So stability is quite important. So the storage of your product, can it be stored at -20, -80? If it's some sort of cell therapy, it needs to be fairly fresh. Do you keep it in the fridge, or how do you look after it? And what is the stability wherever that is kept? And also there's potentially an issue with compatibility. So what container do you want to put this in? Is it a glass vial? Is it the big plastic bags that you might use for production? The closure may have an implication. And also if you're talking about an administration device, i.e. something like an EpiPen, how does that get filled? And is there any bio compatibility issues with that? So essentially all of these documents coming together, these are essentially could be considered as the housekeeping documents for your procedures to get you to the clinic. So these all come together into your CTA submission documents. So that's this workflow again. So these documents really come down into the CMC aspects. So let's quickly talk about CMC. So as I said before, your QTPP is your quality document, and that details everything you want to do from a quality or manufacturing point of view. So once you've got that sorted out and you really know what you need to be doing in the next phases, you might want to think about a CMO selection. So this is contract manufacturing organization. So they will produce your product. So realistically, it may be good to visit the sites and let them really sell their expertise to you. and you need to make sure your product and your style of working fits with their goals. You then move into process development. So this is where it may be useful to come to somewhere like the Catapult, and this is where you move from your small scale lab grade product and you make it fit for purpose to move into the GMP and the CMC arena. And so it might be worth either going to a CMO, a CRO, or somewhere like the Catapult, and we can really help you develop, essentially because this has been done before for other products. So you move towards a more GMP-like product, and at the same time, you start really looking into the methodologies, so the analytical development. So you need methods for characterization of your end product, characterization of any phased products throughout the development. You will need batch release assays, and then you will need some sort of stability determining assay as well. So all these come in at this stage, and you develop those over the course of the next months or years of the development. So the next stage is TT to your CMO. So TT is tech transfer. So you take your GMP quality product, move it to a CMO, and then they can start producing what would be your engineering run. And this is the first of your products where it's almost clinical-like. You can have a C of A, and in theory, this could potentially go into a patient. It's just really about the documentation as to why it may not go into the patient. At the same stage, you do your tech transfer of your methodologies, either to the CMO or a contract research organization that really works in the CMC arena. And then ultimately, your engineering runs and all your batches will be qualified, will be tested with these methodologies. So hopefully this hasn't been too quickly, and you sort of understand the implications of this. But ultimately, you now have your product in a clinical grade, and out of this, you have a data package which includes an IMPD and an IB. You have labeling documentation for your clinical product, and you have your pharmacy manuals to take to the clinic. And so this material here is the material that you will then go forward to put into your pivotal studies. So this is now the safety workflow or non-clinical workflow. So early upfront, you really want to look at a risk assessment, and that is where you really critically look through what data you've got. Are there any ATMP class effects? For instance, if you're talking about an AAV, there may be liver tox, there may be immunogenicity. If you're talking about a CAR T cell, there might be issues of cytokine storm. So this is where you really work out what you need to look for in your safety procedures. And from a gap analysis point of view, you really need to show that you've got robust data to prove the mode of action of your product, or realistically, there's no point in moving forward. So after your risk assessment and gap analysis, it's time to sort of really think about your proof of concept studies. And this is where you prove your mode of action in potentially an in vitro model or an in vivo model. From there, you can then look at efficacy, and this is where you prove your mode of action and your proof of concept within a disease state. So ideally, you want to cure that disease. So for instance, if we're talking about a CAR T again, you may think about a cancer xenograft. So you are targeting the cancer cells with your CAR T's target, and you cure that tumor within that system, and that proves efficacy. So at this stage, you are proving that your product, potentially works to go into the clinic. And you can also think about, the doses required to reduce that tumor by a certain size. Therefore, that can be moved on to the clinical phases. This is potentially a time to think about animal model selection. Your animal models for the proof of concept and efficacy studies they do not necessarily need to be the same as your safety studies. Efficacy is all about efficacy models. So that might be in, rodents, sheep, ostriches. It doesn't really matter. That is the model, the academic model, the gold standard for your product. So make sure the efficacy works in that gold standard product, and then potentially come and talk to us, and we'll show you how to move through the safety studies. So the next stage is to design your pivotal study. This realistically is around about 6 months. You really need to follow the pharmacology of your product. If you have a gene therapy and a transgene, you really want to make sure that transgene works in the species you're talking about. Realistically, monkeys may be the best for this, but obviously that's up to the the ethical committees and a deep investigation of the pharmacology and the the targeting potential of your product. If you're talking about a cell therapy, realistically, you're then going to test it in immunodeficient rodents. If there's no pharmacology, then you may be able to talk to the MHRA or the FDA or whoever about not really doing any in vivo work for this stage, but that has to be justified on a case-by-case basis. And sometimes, also, if there's no pharmacology, it may be worth thinking of a surrogate product. So you make, for instance, a mouse-specific product and test it that way. So you've got your product, well, you've got your efficacy data, you've got your idea of your pivotal study. You can pick a CRO, a bit like you did for the CMO. Go with one that has tested the same sort of products as yours before. They know the route of administration, they know the animal model. Go with those, and then you run your pivotal study. So as I said before, this is around about 6 months, and then 2 to 5 months for any biodistribution, pharmacology, PD endpoints that you may need to justify the safety. And that ultimately gives you your safety parameters to then move into your clinical phase. As I said before, if you look here, it's important to make sure, assuming that you can see my pointer, it's important to make sure that the route of administration that you're testing in these studies is the same as what you want to use in the clinic, and that the test item is handled in a similar way. So ultimately, out of this, you will have a data package which has your study reports and your IB. So as you can see, these all trickle into each other, and there's lots of crosstalk between these different work phases. So now this is the final one, as you'll be happy to hear, and this is all about the clinical phases. So you really want to talk to patients as early as you can up front. So patient engagement, patient advocacy groups, parents of the children that you may want to treat, and really see if they're going to be happy with your therapy. For instance, if the therapy at the moment for them is easy to administrate, and you are telling them that they will need an intracerebral injection or an injection into the back of the eye, they may well not want to sign up for your study. So that is something you may want to think about. So you can talk to key opinion leaders for their input. So that'll be scientists within the space of your indication. So surgeons and patient-facing staff. You want to realistically think about the health economics and market access parameters, and this ultimately tells you, will you get appropriate reimbursement for your product in the future when it finally goes to the clinic? Is there actually a clinical need for this? So therefore, will NICE and the NHS want to use your product? And then also, will it be commercially viable? So will the costs of production, be able to come back to yourselves in the future? So as I said before, the target product profile is the document that essentially grows with your product, and that details everything that you need to then put into the clinical trial protocols and all the other patient-facing or clinical documentations. You need to think about your clinical site feasibility assessments. For instance, if you're talking about a viral vector, can that clinical site even use a viral vector? Is a restriction from that safety point of view. You need to think about the ethics as well as various different issues. And then clinical sites, contracts and agreements to make sure that legally you can do what you want to do within these places. So now we have another CRO, and this time it's your clinical research organization. So these can potentially run all of your studies, or you may want to have your own experts on your own networks, for instance, to recruit your patients. And it's really up to yourselves as to how you go that way. So ultimately, all of this comes to give you your clinical protocol. So the data packages from this are your patient-facing documents, your clinical protocol, and the IB, which you have detailed from the other three work phases as well. So these all come together, as you can see here. So all these three different data packages, the IMPD, IB, all the labeling for the vials and the boxes of test article, the pharmacy manuals, the study reports, the clinical protocol, all come together to go into your CTA submission documents, and that justifies that you, as a team, are essentially fit to go and treat the patients in your first-in-human phase. So the timing of these activities and the decision making is important to ensure that the data packages, really come together to support your CTA. And as I mentioned before, you really need to make sure that you are representative of your intended clinical use throughout your processes and the drug product throughout your process. And essentially, you are proving that this product that is going into humans has been tested, it is as clean and shiny as it can possibly be, and then any implications in the clinical phase you have mitigated for. Okay, so that is the end of this. I hope that wasn't too difficult to comprehend, but anyway, if you have any questions, myself and Nick would be happy to answer them.
Wonderful. Thank you very much, Keith, and to Nick as well, for actually a really good overview with some key examples, so that even if stuff was going over people's head, they had clear examples of CAR T cells and viral vectors that they could latch onto. So I think that was really nice as an overview. Particularly some of our viewers may not have had much exposure to this before. So we have had some questions coming in, and we've had questions ahead of time as well. So if we turn to those now. So the first question is from Stephanie at Oxford University. And she's asked, when hoping to use existing data, so for instance, a previous trial, to support a new trial, how similar do the products need to be? For instance, do you need the same vector but with a different promoter or cDNA? Can it be the same organ but a different delivery route? How easy can you piggyback on previous trial data?
Keith, do you want to take that one? I guess the example might be you have a similar vector route of administration with a different payload, and you want to substitute in another payload. That might be the example here. Keith, do you want to?
Yeah, realistically, I'd like to say that's really easy to cut and paste different vectors, but ultimately, it's not that simple. So speaking of gene therapies, if you have an AAV and it's an AAV1 or an AAV9, and you can say, yeah, there's an AAV9 already out in the clinic. So there's certain things that you can say that AAV9 has proven. So it might be things like the immunogenicity of the vector. So you can say, well, I don't really need to look at the immunogenicity. I don't necessarily need to look at the biodistribution because you have the data to support where it's going to go. But that is to do with the AAV part of that. The main thing you need to worry about is the transgene, so the CDA, the actual cargo. So if it's a new cargo, it will be a new transgene, it will have new pharmacology, and that is going to be completely different. So you may be able to sort of eke back on the animals and say, well, I do not need to do as many biodistribution animals. But realistically, you need to prove that the transgene is doing what you want it to do in the tissues you say it's going to do that to, and that is the toxicology that you have to follow. So you need to prove your transgene is safe at its target area, and it does its job. But also, if you're targeting the liver, and for whatever reason it gets to the lungs, you really want to show that the pharmacology is not going to be have lung toxicity. So it is, slightly more involved than well, not that it needs to be, because it's all about safety, but yeah, the long and end of it is it's fit for purpose for whatever product you're talking about.
Yeah, so there's no skipping steps, but you can use some previous data that's relevant, but that matches what's been done before.
Yeah. If you're talking about a CAR T cell, the latest CAR T cell, so talking about CD19 CAR T cells, the newer products coming to the market, you can see if you look at their documentation that they've done a heck of a lot less in vivo work than the first one's out, because the first one to the market sets precedent, and then you can work on their data and say, well, I don't need to do this because. So if there's a data that you can work around and say this justifies my argument, go for it. But it's difficult because there's only like 6 or 7 AAVs out in the market in Europe anyway to really take that data. Maybe in 10 years' time, when there's 20 or 30, it might be a lot easier. But at the moment, we are all relatively new to this environment, so you really have to do the full package.
Yeah, I think that's very clear. So moving on to the next question from Natalie at King's College London. She would like to better understand the changing regulatory requirements at phase 1 compared to the later phases of going through phase 2 into phase 3, and how they differ.
From a point of view of ATMPs, if we were talking about a small molecule, it's really quite prescriptive. You have to do 2 species to get into first in human. You will do your acute phase safety studies to go into first in human. Then you will do your chronic phases to go into the phase 2, and then you're dipping backwards and forwards between preclinical and the clinic. When you're talking about an ATMP, you really have to do quite long phases in the preclinical space. So we're talking about a 6-month study. So therefore, if you're going into the clinic with a 6-month study, the chronic version of a 6-month study it's difficult. So you can really realistically justify quite a lot in your preclinical space to cover quite a lot in the clinical space. So in which case, if you're clever with your endpoints in the clinic, it may be that you can justify not doing as much work once you get to the clinical phase, because ultimately a test article in a person in the clinic is worth more than in the preclinical phase. So there's lots of scientific rationales about the ethical constraints of doing more work than you need when it's a lot more complicated than the regulations should be, because all the regulations we work to are realistically based on 30 years ago on small molecules and biologics. So it's worth having a discussion with the regulators and really pushing the reasons why your phases can be sort of contracted.
Yeah, so really planning ahead and actually linking up with Catapult, because actually you could give really good advice there as well.
Yeah, exactly. I guess if you pay £1 early on looking into the strategies, that does give you dividends later on in your cascade to get to the market.
Yeah. So if we've got plenty of academics in the audience, if we've got an academic who's developing their product and this is all really new to them, at what point would you want them to, say, contact you and move through this process? Would you want them to have IP secured? Would you want them to have already engaged with patients? At what point should they sort of start feeding in with you, say at Catapult?
Well, we can really help at any stage. My team, the non-clinical team, we can help you right at the start thinking about efficacy models. We can help you with your proof of concept studies, or we can dip straight in and help you with your pivotal studies to support the clinical phase. Or, sometimes if there's something that comes in the clinic and you need to readdress that using a model to look into that safety, we can help you then. So essentially, as soon as you've got a product, as soon as you know what your indication is, you can come to us and we can help you towards that. And Nick can potentially talk about, the process strategies. We can help you with the process of making your products. We can help you with the CMC, interactions, talking to CMOs, and the HEMA, so the health economics. We can help you really work out if your product is, essentially going to make money. And that is what, from an academic point of view, a spin-out company, there's no point in starting a spin-out company if your product is not going to be viable. So, as soon as you've got a product, if you want to, you can come and talk to us and talk to Nick, and likely as not, we can help you.
I think, yeah, just to add to that, I think providing you're translationally orientated, you're kind of not still within that fundamental research space. You've got a mechanistic understanding of how your therapy works. That's always very helpful. From then on, IP is obviously very good if you can file that early. And we would always, we're not IP experts, but think about IP early on. And I'm sure lots of you are versed in this. Maybe worth just delaying that publication a little bit if it allows you to get an IP position, because it will really help you later on if you're going to develop something commercially viable. But yeah, I think providing you have that sort of early proof of concept data, we may be able to engage.
Yeah, that's really encouraging that help is at hand at any point. Because obviously coming from an academic background, like a lot of our viewers today could well be, this process is quite overwhelming. So understanding when to approach different people is quite key.
So perhaps, Carly, just one more point, just because I didn't really cover the process development side of things. I guess, so you've got a cell therapy product. Yeah, if you need help with that process development, providing you have some sort of research grade product where you have something that is like an end product or on the way to an end product, we can work with that research grade product and build it into something a lot more scalable, a lot more robust, taking all the considerations around quality and building up to GMP. Yeah.
Great.
Sorry.
Great. Okay, so moving on to our final question. I think earlier on Keith mentioned an orphan designation. Could you maybe just expand a little bit on that and explain what you mean by that and when that would be applied?
Yeah, so if you're talking about a rare drug, I can't remember the actual definition of a rare drug, but say, one in 20,000 or less.
I think Europe defines it as 1 in 2,000 actually these days.
Wow. Anyway, yeah. So the gist of it is, if it's a a rare illness, some sort of biochemical genetic disposition, the big pharma aren't necessarily going to pump too much money into that unless they know they're going to get something back. So realistically, governments around the world have set up systems where if it's an orphan drug, you're really helped to make it worth everyone's while. So you may have longer covering your IP. You might get sort of help to get to the clinical phases from fees from that point of view. So if it is at all orphan or rare, it is definitely worth potentially talking to our regulatory team, and we can help you push it to make sure that you have protection. And it's really an incentive to get developers to develop the sort of gene therapy, essentially the sort of gene therapies that's what I see gene therapies are for, saving, children that potentially would not have a life because of these sort of rare, these sort of biochemical diseases. So that is the reason for those.
Yeah, quite a key consideration then.
Building on what Keith's saying, is exactly that incentive to give you, I think it can be up to 10 years market exclusivity in the UK or Europe. It might be slightly lower in the US. if you're thinking about commercialization, the timing of orphan drug designation might be also something you want to think about, because there can be interplays between, I think, SME status and orphan drug designation. So, but again, our regulatory affairs team can really guide you on when's best to do that, depending on your goals.
Awesome. So I think that probably wraps up our questions. Thank you very much both for your presentations and for joining us today. Time has really flown by. I just want to share a couple more points before we go today, and that's that session 3 of this seminar series will be happening next week, next Friday on the 3rd of November, and that will look at some of the non-clinical aspects of developing a gene and cell therapy product. And I also want to highlight some of the benefits of becoming a BSGCT member. So obviously these seminars are free for both members and non-members, but there are a lot of initiatives available to members only through BSGCT. So if you take a look through that list there, you can see some of the real benefits of becoming a member. And if you go to the web link at the bottom, you can see many different membership categories as well. So all that's left for me to say is thank you very much for everybody who joined, all of our audience today, particularly those who submitted questions. And a very special thanks again to you, Nick and Keith, for sharing your knowledge and your wealth of experience, giving that great overview today and answering the questions so comprehensively. So thank you again, and goodbye, everybody, and we will see you next week.